Okay, it's live. I just want to catch one thing because it starts screaming at me if I don't click something. There it is. Okay, good evening. Welcome to the Cal State San Marcos Fall 2020 webinar series. I'm Dr. Robert Ife from the Department of Chemistry and Biochemistry, kicking off really the introduction for how tonight's webinar is going to proceed. So there are two ways to interact. If you are part of the Zoom call, you are free to ask questions throughout the webinar. I do suggest holding the questions for after the talk. If you are out on YouTube land, you can also interact and ask our speaker questions by emailing fall.webinar.questions@gmail.com, and I will ask them on your behalf. We've had some pretty good turnout so far. So without further ado, let me hand it off to Amanda to introduce tonight's speaker.
All right, thank you everybody for tuning into this week's webinar. I have the pleasure of introducing Dr. Michelle Arkin. Dr. Arkin obtained her bachelor's degree in chemistry from Bryn Mawr College and continued her studies at Caltech, where she obtained her PhD in chemistry under Dr. Jacqueline Barton. She then completed a postdoctoral fellowship in protein engineering at Genentech prior to her employment at CSUSM Pharmaceuticals, where she was Associate Director of Cell Biology. In 2007, Michelle became the Associate Director of Biology at the Small Molecule Discovery Center and has since become a professor in the Department of Pharmaceutical Chemistry at UC San Francisco. Lastly, as of this past January, Dr. Arkin and several of her colleagues from UCSD and UCSF have founded Algia Therapeutics, a biotech company focused on novel approaches for the treatment of chronic inflammatory and fibrotic diseases. So everybody, I would like to take the time to thank Dr. Arkin for joining us this evening and look forward to a wonderful talk.
Thank you. Thank you, Amanda. Thank you, Robert, for the invitation. It's really a pleasure to be here with you all today. And so today I'm going to give more of a concept-heavy talk instead of a data-heavy talk. Hope that's okay. And I guess the theme of the talk are those obsessions that you have early in your career that stay with you throughout your career, and hopefully they change and grow as your career develops. So, so this, by the way, are my daughter's pumpkins. My daughter Claire is now 14, and you'll be seeing her again later. So I just wanted to start off with a little bit of this theme of career, and I'm going to start with this game. I don't know if anybody on the call or on YouTube has seen the game Careers, but it is a little bit of my holy work to introduce everybody to this amazing game from the mid-20th century that's just totally underappreciated. So in this game, you decide ahead of time what you want out of your life, how much happiness, fame, or money you want. You write it down, and then you go through different careers with that in mind. And so some careers have more fame, but more risk. Some have more money, some have more happiness. And so if I put my own career on this in no particular order, I tried out a few things before I went to graduate school. I worked in art conservation. I worked at the Smithsonian. I worked as a teacher and a counselor in Israel, and then came back to the United States, did my PhD, and then postdoc in Genentech and protein engineering, as Amanda mentioned. And then my postdoctoral advisor left Genentech and started CSUSM Pharmaceuticals, and so I went with him to help start this company. And by the time I left, I started off in biochemistry and biophysics. And by the time I left, I was working in clinical medicine. And now I'm a professor. So the idea of CSUSM and the obsession that drove us at CSUSM Pharmaceuticals was this observation that there are more drugs for some targets than for others. So if you look at all the drugs that we have and you put them on a pie chart, over a quarter of them, some people would say even up to 40%, of drugs target G protein coupled receptors, and another big chunk target nuclear hormone receptors like androgen or estrogen receptor, and another chunk target ion channels. And if you look all the way at the very top where it's very, very small, a couple target protein-protein interactions. And so the question is why? Well, it's not true that protein-protein interactions are rare or that they're not involved in disease. They're ubiquitous and they're involved in lots of diseases. This is a protein we'll come back to called 1433 here shown in blue. And this is a typical signal transduction pathway where you have protein-protein interactions on the outside of the cell, triggering protein-protein interactions through the cell membrane here, triggering protein-protein interactions inside the cell, and then sets of protein-protein interactions that modulate that signal transduction all the way to the protein-protein and protein-DNA interactions in the nucleus. Okay, so it's not rare and it's not uninteresting for disease. So why? What is it that makes protein-protein interactions difficult to target or undruggable? And so the classic view was this. If you look at the compounds that we do, or the targets where we do have a lot of drugs like G protein coupled receptors or enzymes, kinases now, you see that those sites, the binding sites, the active sites of those proteins have deep pockets that a natural ligand fits in. Well, by contrast, proteins interact with each other over a large, broad surface. To put some numbers to that, the average size of a small molecule pocket is about 260 cubic angstroms, where the average size of a protein-protein interface is much larger overall, and it takes place in many little pockets. And each of those little pockets is much smaller than a ligand binding pocket with a natural ligand, small molecule ligand. So then if you try to make a synthetic small molecule, so we want something that's 500 Daltons or smaller, unless it's a natural product. Most drugs are 500 Daltons or smaller, so just 34 or so heavy atoms. Then if you want to make a small molecule fitting into a pocket where the natural ligand is a small molecule, it's easier to conceptualize than a small molecule sitting on this large flat surface where it's just going to roll around. And so we call this the flatness problem. So this woman hanging on the cliff face here is just got tiny little holes, 4 tiny little holes that she's sticking her hands and feet into. And most of her is going towards scaffolding those tiny interactions. So a molecule that's going to bind to a surface like that has to make for example, several very small, very precise interactions, and it has to link those in a very precise way. And that's just difficult to do. But we identified when we were at Synesis Pharmaceuticals that there were exceptions to this and there are ways around this. So even though the structural interface can be very large, here's interleukin-2 binding to one chain of its receptor shown in green. Just the parts of the receptor that bind to the hormone are shown in green. And you can see that it takes up a big chunk of the protein surface to bind the receptor. However, if you mutate different residues at this interface, only 2 or 3 residues play a really large functional role, these shown in red. So we call these hotspot residues. So in other words, the binding energy of this interface is inhomogeneous. There are some areas that are very important for binding and others that are not important for binding. When we did manage to find molecules that bound to the surface, lo and behold, they bound at the same site and they even had the same hotspot residues. So the same residues were critical for binding this small molecule as binding to this protein. The other thing that we found that's really interesting and turns out to be important is that these protein surfaces, even when they look flat in a protein structure, they're actually quite dynamic. So these are different crystal structures, and we're iterating between these different crystal structures with small molecules bound. And you can see that those molecules that bind at that interface trap different conformations of the protein. So even though the protein may look flat, there may be transient pockets that are deep enough to trap with a small molecule. So it's not really a cliff face, but it's more of a molten cliff face. And there are times where you can stick your hand in deeper and get a better hold. So those were 2 things. So hotspots are structural interfaces are larger than functional interfaces. These functional interfaces are adaptive. And the third point is that these functional interfaces are not all the same as each other. Now, over time, we wrote a review in 2014. So for about 10 years, there had been progress made on protein-protein interaction inhibitors, drug-like inhibitors. And there were enough that we could start to find some principles about what are good targets, which targets are more likely to bind small molecules than others. And we divided protein-protein interfaces into 3 sections: primary, secondary, and tertiary. And the idea of a primary interface is that the interaction is made up of the primary sequence of one of the parts of the protein interface. So this protein, here we have a globular protein with a hole in it, and the other protein, a histone, all of the binding affinity is governed by just one or two residues. So just the primary sequence of that histone. Secondary interfaces, I think you can guess what's coming, require the secondary structure of the other protein to recapitulate most of the binding affinity of that interface. So you could call this a hot segment. If different residues are hot spot residues, these are hot segments. You can cut them out of the protein and maintain most of the protein-protein interaction energy. And then there are tertiary interfaces. These, like IL-2, are the hardest and the most difficult to design because they require the entire tertiary structure of both sides of the interface. And perhaps not surprisingly, the more complex the surface is, the fewer drug-like inhibitors there are. And the simpler the surface, the more drug-like inhibitors have been discovered. So taking this knowledge and being at Synesis for about 9 years, we discovered many inhibitors of protein-protein interactions. Some of them went further than others. This went all the way to market. It's now marketed as Zidra, sold by Novartis. That's a story in and of itself, how a drug starts at a startup company and then it goes through multiple different companies before it's finally marketed. And then, as Amanda mentioned, I eventually became the Associate Director of Cell Biology, and there we were working on clinical stage compounds, trying to understand the mechanisms of action, selecting patients, identifying biomarkers. And 2 programs I worked on in the clinic. One was a CDK inhibitor, and the other vosaroxin, shown here, which went all the way through phase 3 clinical trials. It has not yet been approved for use. So I had kind of started off in biophysics and structural biology, the things that I showed you, and then I wandered all the way down from discovery through translational and clinical development. And what I realized when I got there was that it's really great to know what a molecule has to look like and what biology has to look like to be clinically viable, but actually my heart was here in discovery. And so I left the company I helped start and moved to UCSF. So now what do I do as a professor of pharmaceutical chemistry? Well, I run a chemical biology lab. So we use small molecules and protein engineering tools to characterize, to discover novel molecules and then use those to probe new biology. I also co-direct the Small Molecule Discovery Center, which is a drug discovery and chemical biology center that many people in the community can work with. We do high throughput screening and other types of drug discovery activities. High content imaging, I can talk some more about those later. So those are my main jobs, but then, you know, professors, there are a lot of opportunities to get distracted. I teach, I travel to conferences, I talk to good people like you. And there are a lot of opportunities for leadership also. I sit on boards for companies. I also sit on the boards of professional societies. I'm the president of the Academic Drug Discovery Consortium, for example, which is a professional society. So those are some of the activities. What are the perks? Well, for me, the perks of this kind of job are being connected to a broad swath of research. As the director of the Small Molecule Discovery Center, I sit in my office and just brilliant people come in my office all day long asking me for help on their cool new projects. So it's very interesting to see what's going on. And then as a professor, I can build programs that I'm really passionate about, that are important to me. And those obsessions that I mentioned. And then also because as a faculty member, you have a lot of ability, a lot of flexibility in what happens to the programs that you develop. And so we're very involved in licensing and spinning out companies. Amanda mentioned Algea Pharmaceuticals that we started in January. And I'll talk about the work that led to another company called Ambigon that started in July. So what are the biggest challenges of a job like this? Well, there are the flip sides of the perks. One is that if we really want to do drug discovery, it's a huge interdisciplinary process. It's very expensive and takes a very long time. So there's a real disconnect between funding models and really the way a lab is set up in academics and the costs and timelines of translational research. So now we're really have a focus, certain part of our lab is really focused on how to address this. In particular, how can we use artificial intelligence and really high performance computing models to cut the time and cost so much that those brilliant people that come in my office and want to do drug discovery, we can actually help them take those molecules further than we can now. And also deciding where to focus amid so many opportunities. So really trying to keep track of what we should be doing. Where can we have the biggest impact? So I'm going to talk the rest of the time about building a program that I'm passionate about. And hopefully you'll see some of the themes that started at Sunesis and Genentech and that we're developing further. So I already told you about protein-protein interactions and the flatness problem. And then I also told you that it's not as hopeless as it seems because there are regions of flexibility that create dynamic pockets. So there's the cliff problem, but on the other side, there's also the disorder problem. So some protein-protein interactions, far from being rigid like cliff face, are very flexible and dynamic, and the proteins themselves are not well folded into a single structure until they interact with another protein. So here, when they're in their unbound state, they're like spaghetti. And how can you design something that sticks to a protein with no structure? Even the spaghetti sauce doesn't stick to the spaghetti. Okay, so we have the flatness problem and we have the disorder problem. And then we have the big Megillah problem, which is the complexity problem. So we talk about these protein-protein interactions, and I talked about primary, tertiary, secondary interfaces. It's as if there were two proteins interacting alone on a dance floor like this waltzing couple, where really biology is more like this English country dance, where you have a complex set of interactions that happen in a specific sequence. And if you want to watch that dance and that dance gets deranged in some way, the music changes or somebody drops their guitar, then the dance is messed up. And if you want to bring that dance back into alignment, you need to understand the entire dance. And furthermore, if part of the dance is not working out so well, like you don't want this guy to dance with his ex-girlfriend, you don't necessarily want to take him out of the running altogether. You just want to block this one interaction. So looking at that in a little bit more serious way, here's an example of a subset, just a little piece of a protein-protein interaction network. And the reds and blues are telling you which parts of the network are amplified or inhibited in different disease states. These are different cancer models. So here we have a number of proteins interacting with each other. Let's just take this one. This protein happens to be, this is the gene for the beta isoform of 1433. And you see that this protein is interacting along edges with a number of other proteins. So each circle, a node, is a protein, and each line between them is an interaction edge. So if I wanted to understand how this protein worked and how it worked in disease, so for example, it binds less to this protein, this is a CDK antagonist, and it binds more to this protein, it's the DNA repair factor. In the disease state, maybe I want to block this or amplify this, and then I want to leave this one alone because it's not altered in the disease state. So the overall question is, how can I specifically modulate just one interaction edge instead of a whole node? If you inhibit an enzyme function, you might knock out a node. If you inhibit expression of the protein, you knock out a whole node. You can see what a much larger perturbation that will have on the system. So going to that protein, 1433, we chose 1433 as our first foray into this question about really how to specifically modulate one edge of a complex network. So we picked maybe the most complex network. 1433 binds to phosphoproteins and it alters their fate. So when a protein is phosphorylated, you've probably seen lots of signal transduction pathways like the one I showed before. In signal transduction, proteins are phosphorylated and then they phosphorylate the next protein and so forth. And eventually it ends usually in transcription factors or in proteins being degraded. So why do proteins get phosphorylated? What happens to them when they're phosphorylated? Well, sometimes they're activated, they change conformation and they're activated. But a lot of times the reason a protein is phosphorylated is that it makes it a target for 1433. The 1433 is like the taxi cab. The protein gets phosphorylated, that's like raising its hand. The taxi cab comes and picks up that protein and takes it somewhere. but where it goes depends on the protein, not on 1433, but on the client protein. So depending on where that phosphorylation site is, the protein may be activated, it may be inhibited, it may be degraded. So a vast array of biological fates occur in a 1433 dependent way. And hundreds of proteins interact with 1433, including lots of high-value undruggable targets, like transcription factors. Lots of spaghetti types of targets bind to 1433. So here's the structure of 1433. It's a W-shaped dimer, and the phosphoproteins bind in this groove. So if we turn that 90 degrees, now 1433 is shown in white, and an example client protein, or the peptide derived from a client protein, is bound across this groove. Here's the phosphorylation site, the phosphoserine site. And then proteins, as you'll see, vary significantly in their sequence identity and also in their conformation, the conformation in which they bind in this primary groove. But this site is very consistent. So this is the work of my collaborator and friend, Christian Altman. And his obsession, his career-long obsession, is asking this question: how can we take this 1433 network and get selectivity for specific interactions in that complex network by stabilizing their interaction? So by stabilizing the interaction of the client with 1433, how can we amplify 1433 mediated biology? So this might sound crazy. How are you going to develop a molecule that stabilizes a protein-protein interaction? But actually, that's what natural products tend to do. Rather than inhibiting them, they tend to stabilize protein-protein interactions. And these are some very famous molecules. Fusacoccin binds 1433. Rapamycin, you're probably familiar with. It binds to FKBP, FRB. It stabilizes a protein interaction that would not normally occur. So rapamycin stabilizes a non-native interaction. These proteins only interact in the presence of rapamycin. And the function of rapamycin binding is to inhibit the activity of the protein that it's binding to. Now, fusacoccin, on the other hand, binds to 1433, stabilizes its interactions with its native substrates. This is in plants in particular. And that activates the function of 1433. And in fact, this particular interaction activates the function of this ion channel, and then the ion channel can't shut off, and it kills the plant. So we have both inhibitory and activating functions. We have the ability to target non-native interactions or native interactions. So by stabilizing a protein-protein interaction, you could imagine higher selectivity and higher functional variability, flexibility. So I said that the network is so complicated. What makes us think that we can target it selectively? Well, we're crazy, that's why. But if you look at the protein-protein interactions from crystallography, you can see what I was telling you about before, that the sequences, here are 3 examples, and their structures vary considerably in this primary binding groove. So estrogen receptor, for example, is phosphorylated at the second to last residue. This is the C-terminal residue of the whole protein, and it leaves half of this primary binding groove empty. P53 interacts with 1433 near its C-terminus also, but instead of going across the groove or stopping here, it turns out of the groove, leaving a different shape and a different sequence identity, so different shape and different functional moieties in this primary binding groove. YAP and TAZ, transcription factors in the Hippo pathway, bind across the entire groove, filling this particular site. And the reason I keep pointing out this site is that this is the site that fusacoccin binds. So here, fusacoccin, it's a natural product. As I said, it kills plants, but it also binds to a handful of human proteins. So this is human 1433 and human estrogen receptor. And fusacoccin binds in this groove. It basically creates a new wall in this binding groove and stabilizes this interaction by 1,000-fold. So this can be quite a significant effect on this protein-protein interaction. We also know from years of work from Christian and his colleagues that varying the structure of this natural product allows us to vary which targets 1433 is bound to. So which complexes are stabilized by these different natural products depends on the structure of those natural products. And when you stabilize different interactions, you get different biology. So fusacoccin itself stabilizes and activates the interaction, stabilizes the interaction between 14-3 and the cystic fibrosis transmembrane conductance regulator, CFTR. This is when it's mutated, it causes cystic fibrosis. 14-3 stabilizes the ion channel and helps traffic it to the surface. So if you increase the functional activity of 14-3 by adding fusacoccin, you see an increase in functional ion channel at the surface. Other analogs can inhibit signal transduction and have an anti-cancer effect, or they can stabilize regenerative processes and have a regenerative or pro-growth effect here in axons and neurons. So a wide array of biology depending on the selectivity of the interaction. But fusacoccin binds in that one site, and it's not that easy to change, and it always binds in that site. So in doing lots of work and different strategies that we've taken to try to find compounds that we can say approaches that we can use to stabilize any interaction with 14-3, we found that fragment-based drug discovery is the most flexible. So if we take those 3 targets again that I showed you before, fusacoccin doesn't bind to all of them, but we can identify fragments that bind to and stabilize each of these interactions by binding in different subsites at this binding interface. So we're going to use a fragment-based drug discovery approach. So what do I mean by that? If you look at a peptide or a high-throughput screening library or a natural product, you can break down those molecules into subsets of the molecule. So components that strung together make up the structure and function of that molecule. When we use high-throughput screening, when we use a functional assay, we'll take hundreds of thousands to millions of these molecules, and we'll test them for changing the function or for binding to a protein of interest. The chances, if that protein of interest has a complex surface, so remember I said protein-protein interactions tend to have 3 to 5 subsites that are very specifically separated from each other. The chances of making a molecule a priori that's going to satisfy all of those interactions and not make any bad interactions becomes very, very unlikely. And so when we screen 1 million compounds, we tend not to find anything that binds productively to the surface. If instead we break the compound up into little pieces and we just screen those little pieces, which we call fragments, the chances of finding a fragment that binds in one subsite is much higher. And then we can link those 2 fragments together in a subsequent step to get a high affinity compound. Okay? So a fragment is a molecule that's about 1/3 to half of the size of a drug molecule. And it's been very successful for difficult targets for the reasons that I just said, that you've got these multiple distant subsites finding her instead of just finding her hands, much easier to just find her pieces and then link them together later. So our goal for this project is to develop fragment-based approaches to systematically, that's really the key word, systematically, proactively stabilize specific 14-3 client complexes to inhibit disease processes. And so we're starting with cancer, but there are many disease processes that we could expand into. So in a cartoon fashion, we have 14-3 dimer bound here. We have a protein that binds to it. And then we have a small molecule that acts like gum or 2-sided tape that fits in this rim of the interface, contacts both 14-3 and the client complex to stabilize this interaction. Now, the parts of the phosphorylated protein that bind to 14-3 are disordered. They're disordered regions of structure until they bind to 14-3. So here we have a solution for the spaghetti problem. When the spaghetti binds to a specific site, it becomes ordered. And now we have an ordered structure here that we can target with a small molecule. So what technology are we going to use? So we're actually going back to a technology that we used at Synesis Pharmaceuticals, was discovered there. And we brought it with us when Jim Wells, the founder of Synesis, and I moved to UCSF. It's called site-directed fragment discovery, or tethering, disulfide trapping. So in this technology, you have a protein with a putative binding site and a protein of interest, a cysteine residue near that binding site of interest. Now, this cysteine residue can be native, wild type, or can be engineered. if you're looking to go to a specific site. We have a library of about 2,000 now disulfide compounds that are the same here, but they differ on this end. And this is the binding element. We mix them together with the protein under conditions that favor exchange between the thiolate, the thiolate and the disulfide. And at thermodynamic equilibrium, we'll find compounds that can make this disulfide bond and also bind to that protein of interest. Okay, so binding is required for us to trap this fragment complex. And we detect that by mass spectrometry. So here's an example of a dimeric protein. The cysteine residues on the large subunit don't bind to this particular fragment, but we can see that the small subunit has the wild type mass, and it also has the mass of the adduct of the disulfide trapped fragment plus the protein mass. So there are many advantages of this technique for stabilizing protein-protein interactions, we thought. One is that it's a site-directed approach. So we choose what site. So we can say we want to find something that binds in the fusicoccin site, or specifically something that doesn't bind in that site, that binds in a different site. Second, most fragment-based screens are based on binding. And that's because those molecules are really small. There's not a lot of energy that they have for binding. And so their binding affinities tend to be weak. And it tends to be easier to find binders, find active molecules when you find them for binding. And then you have to optimize them to have enough potency, enough binding energy to affect function. But here, we can tune this disulfide strength by tuning the reduction potential. So we can make this disulfide bond stronger or weaker, and that allows us to saturate the binding site. So then we can screen for function in the primary screen. So I'll show you how that's done. And then just to show you a little bit about what this library looks like, the library has a cap that has half of the disulfide on it and a solubilizing group. Then the disulfide is linked to the diversity element or the fragment by a series of linkers. And these linkers vary in their geometry and in their length. This is an important part of the application. So the fragment needs to bind, it needs to adhere like this ball, needs to be complementary to the surface. But the distance and geometry between that ball and the cysteine residue is equally important for finding the fragment. So the target that we're going to start with is estrogen receptor. And estrogen receptor is inhibited by 14-3-3 binding. So this is a potentially good cancer target. So this is how it works. Estrogen receptor is bound to a chaperone, HSP90. It's not a stable folded protein in the absence of estradiol. Then when estradiol comes into the cell, it binds to the ligand binding domain of estrogen receptor and liberates it from HSP90, changes its conformation, which allows it to dimerize. And now it has a choice. If it dimerizes, it can enter the nucleus, bind to its transcriptional elements and mediate transcription, which can cause breast cancer. If this conformational change, this conformational change also exposes the C-terminus of the protein, which allows it to be phosphorylated. So if it becomes phosphorylated and then binds to 14-3-3, this antagonizes its nuclear entry. It sits in the cytoplasm as an inactive monomer or dimer. We don't know yet. And so this is just a little piece of data to show you what I mean. So as you titrate in estradiol into a cell line, you see an increase in a reporter activity. That's in black. If instead, before you add estradiol, you add fusicoccin, fusicoccin stabilizes the interaction between estrogen receptor and 14-3-3, and it significantly blunts the activity of estradiol. So you get much less transcription. And actually, this is published data, when we do the experiment, we can totally block transcription. If you mutate estrogen receptor so that it no longer binds 14-3-3, now fusicoccin has no effect. And in fact, you have higher transcriptional activity because this is a native antagonistic interaction that's going on in the background all the time. So that's how fusicoccin works. So here's a close-up of that site again, 14-3-3 shown in white, estrogen receptor peptide, the C-terminal peptide from estrogen receptor shown in green. This is just the C-terminal residue. This is where fusicoccin binds shown in yellow. So we wanted to see if we could use the disulfide trapping approach to find stabilizers that bound in the fusicoccin site. So we have one wild type cysteine residue here at the distant periphery of the site. We weren't sure if that was too far away, so we also mutated that out and mutated cysteine residues in at the I+4 and I+7 residues. So moving along an alpha helix here towards the ER alpha binding site. And we're going to do that disulfide trapping experiment with one of those cysteine residues mixed with our library, either in the absence of estrogen receptor peptide or in the presence of estrogen receptor peptide. And we're looking for molecules that bind better in the presence of estrogen receptor than in the absence. Because if the peptide stabilizes the compound, then the compound will stabilize the peptide binding. So you see how that allows us, that's a functional screen. We're screening for compounds that bind better in the presence of peptide than in the absence. And the Goldilocks residue for this particular screen was the one in the middle, was the 42C mutant. Each dot here is a compound that we tested. We tested in the absence of peptide and in the presence of peptide. So if you look at this particular dot, this compound was about 70% labeled, bound to the protein, about 70% in the absence of peptide and about 70% in the presence of peptide. So it's not stabilized by peptide binding. Compound 1, on the other hand, goes from about 25% bound to about 60% bound. So that is binding cooperatively. So now we characterize these better. We flip the experiment around a little bit. Now, instead of looking at compound binding, we're looking at peptide binding as we titrate in compound. So here we have an estrogen receptor peptide that is labeled with a fluorophore, and we can detect the apparent mass of that peptide, of that fluorophore, based on whether we have fluorescence anisotropy or not. So it has low anisotropy when it's unbound, high anisotropy when it's bound to the protein. And we measure that here. So we start at a concentration of peptide where we have very little binding. As we titrate in the compound, you can see the compound is inducing the peptide to bind with an EC50 for the compound of about 2 micromolar. We can then do the experiment in a slightly different way where we ask, what is the apparent binding affinity of the peptide in the presence and absence of the compound? And we can see that the apparent affinity of the peptide is about 2 micromolar in the absence of compounds, and it's increased in its binding affinity about 40-fold when we saturate the binding site with compound. Crystallography, this is all done in Christian's lab. Crystallography shows how the compound binds and stabilizes the interaction. It binds in a little pocket in the back of the 1433 and makes connections with the C-terminal valine residue. And that stabilizes this peptide binding. So we've looked at that some more, and we've done a lot of chemistry, but I just want to talk about one other concept first. And the question is, okay, there the cysteine residue was on 1433. What about clients that contain a cysteine residue? Isn't that another way that you might get selectivity? So here is estrogen receptor related receptor gamma, ERR gamma, and ERR gamma has a cysteine residue, you can see back here, in the +1 position, so the residue next to the phosphorylated serine. This is the binding site of fusacoccin and of our original molecules. And so you see that that cysteine residue is pointing towards the same region of 1433. So we asked whether we could do a screen starting from the serine on the peptide. And so that screen we did differently. Here we took the peptide, the ERR gamma peptide now, with a fluorophore on it and its cysteine, under conditions where it's not bound very much, so it has low fluorescence anisotropy. We then added the compounds one at a time and measured whether the binding of the compound increased, whether the compound bound, and whether binding of that compound increased the fluorescence anisotropy of the peptide. So we're measuring peptide binding in this screen. And it turns out we found compounds that did stabilize. Here's the ERR gamma peptide again in orange. And here's a crystal structure showing the disulfide bond to the fragment in teal that we identified. This fragment stabilizes the peptide binding by about 5-fold. If you see how it binds compared to the fragment that I showed you from the estrogen receptor project, it binds in the same location. So the linkers come from different places, depending on whether they're bound to protein or to peptide, but the site that they find on 1433, that stabilizes the interactions with peptides is identical. So in other words, we were talking about protein-protein interaction hotspots for protein-protein interactions, but here for protein small molecule interactions, we also use disulfide trapping to identify a hotspot. So what do we do next with these? So there are many different ways that we can optimize the compounds now so that they have the potency and selectivity to test in cells or to turn into drug leads. So starting from here, we have 3 ways we can go. There's an approach called extended tethering, where we put a thiol on the first fragment and then screen for a second fragment. There's an approach where we just simply take the first fragment and we take this highly dynamic disulfide bond, unstable disulfide bond, and we convert this molecule into something that will alkylate the protein, making a covalent stable bond. This we can only do if we have a wild type cysteine residue, not an engineered cysteine residue. And then the second, this was the original idea that I told you about when I introduced fragments, that you take the first fragment, then you find a second fragment that binds nearby, and you link them together. And so just pulling data from the presentation, I'll put them on the same figure, and you can see that one of our estrogen receptor alpha fragments binds right next to a fragment from another fragment screen. So you can imagine how you might link those two together to accomplish this improvement in potency. So we've done this screen several times now with different protein peptide interactions, and this is just an overlay of a few examples of crystal structures of 1433 in white and different client proteins shown in gray. So what I'm not showing you here is this the serine or threonine phosphorylation site. That's right here where all the peptides look very similar. And you see the diversity in structure on both sides of that phosphorylated residue. So we can take several different peptides here and using the same sequences, identify different fragments that bind and stabilize in this site. So we've stabilized peptides with different binding modes and different sequences. So our next stop is chemistry and cell biology. So in the next couple of minutes, I just want to come away from one set of obsessions, which is how do proteins interact with each other and how can we modulate that in a specific way, to more globally, why is there so much failure in drug discovery? Part of it is that we know, for example, protein-protein interaction is important, but we don't know how to drug it. What are some of the other reasons that we have so much failure in drug discovery, such that 1% of the projects we think are worth doing end up with a drug at the end? And most of them fail in the clinic in phase 2 clinical trials. Even after all the work that goes into selecting a molecule and selecting a biology to put into clinical trials, 50 to 100%, depending on the therapeutic area, of molecules will fail at this stage. And this is the first stage that we're really testing for efficacy, that we're testing our drug in a human population with the disease, and we fail most of the time. So why is that? And what can we do as academics up at the very top to make this funnel wider so that fewer things fail at this expensive stage and where we're using humans in our failure? So we think about some of the different modes, why things fail and what we can do about it. So in broad categories, for you to think about in your own research, we select the wrong targets. We select the wrong targets either because we know what the right target is, but we don't know how to make a drug for it, or because we don't really understand biology well enough to identify the correct targets. Second, even if we have a pretty good handle on the biology, we select the wrong model systems to do the screen. And there's an adage that you get what you screen for, and that's really true. So if you simplify your screening assay too much, that maybe it's fast, maybe it's cheap, but it's not biologically relevant, the molecules that you discover may never become biologically relevant. So in our lab, we work hard to develop models that are as complicated as they need to be to recapitulate the biology as we understand it. And finally, and this is kind of a subtle thing to try to share, but we focus on affinity instead of on the therapeutic index. So we focus on making a molecule bind really tightly, and then much later in the process, down here in IND enabling studies, we really focus on the safety of that molecule. And sometimes we end up with molecules that are very potent at their target, but they're very potent at an off target also. and they're too toxic. So in addition to fragment-based drug discovery in our lab, we look at a number of other ways to approach these novel targets to try to address the undruggability, the model system problem, and the therapeutic index problem. So I mentioned how protein-protein interactions have regions that are highly dynamic. That's true of enzymes also. So this is my favorite molecular machine called P97, and you can see how it moves when it turns over ATP. And you can imagine how small molecules might be able to trap different conformational states and how that might give you different biology. We can also take advantage of the cellular context by using primary cell models or complex cell cultures or even whole organisms when we work with parasites. We can get more biologically relevant systems to do our initial small molecule discovery. And then finally, to address this question of therapeutic index, we can try to optimize several parameters at a time. And one of the emerging ways to do that is to use high performance computing and artificial intelligence to ask of our computer to find molecules that don't hit this target, but do hit this target and are soluble and don't hit this ion channel and all kinds of other things. So we'll give it multiple properties that it needs to optimize in parallel. So with that, I just want to thank the people that worked on Team 1433. The project was started by Alina Sebesma when she was a master's student in my lab. She was a graduate student in Christian Altman and Luke Brunsfeld's lab. She was working with Ken Hollenbeck, graduate student in my lab. And now the project has got several excellent chemists in the form of Priya Shankar, a number of collaborators. Bente Somsen worked on the ERR Gamma project. Thanks to Ona Pharma Foundation for a breakthrough award for this work, and our co-founders to form Ambagon Therapeutics to identify drugs from this technology. So finally, I started off talking about careers and how we have lots of careers and we try to decide ahead of time what our values are. But I think you can see from my own career going from pharma to academics and now starting companies that you can really invent your own career. And so this is my daughter when she was 8 or 9. One Christmas break, we decided to make our own game. So we took the careers game and we turned it into a new game called Hit the Road, about travel. And so I encourage you to hit the road and invent your own exciting career. And thank you very much. Thanks to my lab, and thank you for your attention.
Right.
Thank you, everybody.
Talk. Thank you so much for that. And then at this point, I'd like to invite Dr. Ike to give some rules about how the questioning will be handled.
Oh, yes. Thanks, Amanda. So first off, Michelle, fantastic talk. Thank you so much. And this last bowling picture, man, I really want to go bowling right now. This is not fair. But if you are out in YouTube land and you have a question, if you're in Chem 440 and you have a question, please feel free to email me at fall.webinar.questions@gmail.com, and I will ask them on your behalf. But otherwise, Amanda will call on questions.
All right. So do we have any initial questions for Michelle? In the meantime, I do have a question. Do you do the drug synthesis, like the small molecule synthesis in your lab, or do you have like a different group that works on that for you, or are they already like known molecules that you're just testing?
Yeah, we do a lot of synthesis, and it's kind of an evolving question. So the Small Molecule Discovery Center has a chemistry group, and it used to be headed by an amazing medicinal chemist named Adam Renslow. And so he did a lot of this work with me. And now our associate director of chemistry, Jeff Neitz, works on the project with us too. And so we have postdocs and specialists and professional staff, as well as students and postdocs. And then sometimes we outsource work too. So if we want to make a whole bunch of compounds that are really simple, then we'll have a contract lab do them. So it's a combination.
Cool. Thank you. Let's see. Tamara has a question. Go ahead, Tamara.
Hi, thank you so much for your talk. It was very interesting. I had a
Thank you,
question from earlier on in your talk when you were at CSUSM, what you were doing. This is going to be probably a very poorly worded question, so just bear with me. I'm trying to process it as but as you've kind of gone through the process of drug discovery and creating certain types of drugs, have you found a pattern, or have you noticed that certain diseases or certain targets kind of lend themselves to protein-protein interactions, like for creating new drugs for them? Yeah, so the Zedra drug or the other ones on that list, were
Yeah.
particular kind of thing that you were looking for, or was it? I know protein-protein interactions are ubiquitous, so what commonality?
Yeah, no, yeah, it's a good question, and there are a few ways to answer it. And so hopefully one of them is the question you were actually asking, but if not, ask again. So if you look at those networks, for example, those networks have, so probably like slide 4 or something. Yeah, some of these are enzymes. And some of them are protein-protein interactions, and some of them they're all kinds of different functions. So how do I decide when I want to target a protein-protein interaction and when I want to target an enzyme? Or when I want to degrade the protein? Maybe you've heard of PROTACs. And so these are ligands that bring the protein into proximity with the degradation machinery. So we have a lot of different ways that we can try to manipulate the biology. And we don't get extra credit for doing the hardest one, unfortunately. I'm always drawn to the hardest one. But so you really want to think about what biology are you really trying to affect? What are the implications of targeting it in that way? So if I target, for example, RAF is a kinase. If I inhibit kinase activity, I'm going to inhibit all of the different enzymes, all of the different substrates. If I stabilize or inhibit just its interaction with 1433, then I may cause a different biology. It may be more subtle, but it may be safer. So you really need to understand your biology, and then also your modality. So if I want to degrade the protein entirely, I'll really knock out all of its functions, its scaffolding functions, and its enzymological functions. So is that what I need to do? And what are the kinetics for doing that? Or maybe I want to target outside the cell. And if I'm targeting outside the cell, then I can use an antibody or a protein therapeutic as well. So all of these things have this ripple effect, how you're going to do the screen, what types of molecules you're going to develop. So it's really about understanding your biology.
Thank you.
You're welcome.
All right. Now Eric has a question.
Thank you for your talk. It was great. My question is, would you attribute the lack of protein-protein interaction drugs that you had on your pie chart from before due to increased complexity needed to develop drugs for those compared to small molecule drugs, or due to a lack of maybe instrumentation techniques such as the screening methods you talked about with some of those AI programs? And do you feel that with those screening programs that we'll see an increased number of protein-protein interaction drugs in the future?
Yeah, it's a good question, and it's always hard to predict the future. But if I go back a little bit to the past, to so I mentioned in 2014 we did this review. So there were enough projects then in the intervening 10 years that you could start to see progress. And what you see are these waves. Some technology comes out, or some biology is discovered, some new modality of drug comes out that reinvigorates one's interest in a particular target class. So a beautiful recent example of that is RAS. The KRAS is a potent oncogene. People have wanted to inhibit oncogenic KRAS for 40 years. Failed for one reason, try again, failed for another reason. Then what really the most recent successes there have actually come from the disulfide trapping technology. My colleague Kayvon Shokat said, well, what if I just try to target an oncogenic cysteine residue? Can I use disulfide trapping to find a compound that binds just in an allele specific way to that mutant? And he did, and developed potent compounds, started companies, and now there are 2 or 3 compounds targeting KRAS, once thought to be impossible, in the clinic. So that was a technology, and thinking about the technology in a new way. So I think that's a paradigm. There are many different examples. Gene therapy comes back around again and again. Antibody drug conjugates come back again and again. So it's the linkage. of technology and biology.
Thank you.
And stubbornness is the
Wow.
magic ingredient.
Usually it's. I've got some more questions from Roberto.
Hi, Dr. Arkin. I want to say thank you for that amazing talk. It was super interesting. I have a couple questions. I'll ask like
Thank you.
Tinao and wait if anybody else has any questions. But my first one was about 1433. I guess it's like a really simple question. Is it considered like a chaperone? Because like I remember learning
Yeah,
about chaperone protein. I'm like, is this considered a chaperone then?
sometimes I call it a chaperone. I think it's probably not technically the right term. Probably an adapter protein is the most general term, because sometimes it does stabilize protein structure, and in that way it would be a chaperone. Sometimes it brings to, so this is a dimer, right? So sometimes you can make it dimerize. It can be a scaffold for oligomerizing proteins, and then you might call it a chaperone. But then in other cases, it leads to its degradation or inhibition. So yeah, it's too specific a term. But yeah, chaperones are really, really cool.
Okay. Okay, cool. Interesting. So I remember when we were talking about the small molecules, you were talking about that we were using for the sulfide, that you were using for the sulfide bridging.
Right.
You mentioned something about controlling, I'm not sure if you said reduction or oxidation potential. I guess my question was, how do you do that? Like, how do you control that?
Yeah, redox is what I said. So both reduction and oxidation.
Uh-huh.
So there are two things that you can control here when you do this experiment. I mean, you can control concentrations and things, but the amount of reductant that you have, if you have a high reduction potential, so you add a lot of reductant, like free thiol, then it's going to stabilize the thiol state rather than the disulfide state, which is the oxidized state. If you have too little reductant, then you'll favor this oxidized state. So you want to tune that system so that you get an exchange back and forth. And it's not really a thiol. I should draw it as a thiolate. So it's really the S minus, and that's governed by pH. So the thermodynamics won't be changed at different pHs, but the kinetics will be changed. So the more basic it is, the more reactive this thiolate will be. And then you tune the amount of beta-mercaptoethanol or glutathione, some common reductant, to get the right balance for this particular cysteine.
Cool.
Does that help?
Thank you.
Okay.
Yeah.
Sure.
Thank you.
Thanks, Roberto.
There are questions, so I'll let you two people ask some questions.
All right, like Roberto mentioned, we have a few questions from YouTube land.
Okay, so I do have a few questions. So the first one is actually, I think, a fun question. Michelle, are you a rock climber? Somebody wants to know.
Yeah, I should. If I could find somebody who wasn't, I think she's Thai, so clearly not me. Yeah, no.
The second question is based on the disulfide trapping, and I think this slide works very well. Since the tethering can come from multiple different areas on the protein, do you have to attach the potentially active small molecules in different ways on the tethers?
Yeah, so that's there are a couple of reasons that's an important question. So two sides. One way you could say, well, there might be lots of cysteines on the protein, right? What about other cysteines on the surface? Do you have to remove those? And the whole, like I mentioned, there's a lot of reductant here. We have beta-mercaptoethanol, so we have this kind of stuff. So it's a competition-based experiment. So I like to have other cysteines on the surface. As long as they're not near pockets, I won't find anything there. And then the question about binding pocket. So unfortunately, I didn't put the slide in here, but remember I did the screen for estrogen receptor with 3 different cysteine residues on 1433, looking for stabilizers. And what you find is just the same thing that we found with ERR gamma and ER alpha, that fragments coming from different places go to the same binding site. So there's a binding hotspot, and what varies is the linker. To find, it's a fishing pole. So you've got the fish in one place, and where you stand on the dock, you need different type of fishing pole to get to it.
Fantastic. And then a third follow-up question from one of our masters in biotechnology here at Cal State San Marcos. And this is kind of a broader question. How has academia prepared you for running startups? And then how did the first get started, or how did you first get started in working in startups?
Yeah, so I got started at the startup company because my boss left Genentech after 18 years to start this company, and I had just started my postdoc. I'd been there for a year. So I just decided, what the heck, why not, see what it's like. It was good investors, good people. I was enjoying the work. So serendipitously is how I got in there. And academics, I think the combination of having been at a startup company. So first of all, I think being at a startup company helps with being an academic. And so I think that goes both ways, because both of those jobs are really kind of seat of your pants kind of jobs. They're very entrepreneurial. Like, I'm always having to raise money. I'm always having to figure out who my audience is, how to convince them to invest in me or invest in my ideas. So it's the same thing whether you're at a company or in an academic lab in that sense. And then you also have to be scrappy. And the best companies and the best labs are really mission driven. So everybody's pulling in the same direction. Everybody has the same big goal, and everybody knows that the success of the person next to them is their success. So that's not always endemic in academia, but it could be. And so that's something I think that I bring to my lab, having worked at a company.
That's a unique point of view. I like that.
Thank you.
And thank you, YouTube. If there's any more questions, please put them in the email. On to you, Amanda.
All right. I've got another question from a master student, Lisa.
Hi. Thank you for your talk. So my question was, I believe you said in the beginning that you had started out in art. So I was wondering how you transitioned from art to science.
So I took art history when I was a freshman. I didn't have to take English, so I took art history instead. And I was taking chemistry, and I really liked it. And then I found out that there was this new kind of field growing up called art conservation science. And this was about using physics to understand the work of art. So using the same kind of tools that I use now, even in protein chemistry, to understand how proteins work, applying those to how paintings worked, how their colors had changed, or looking beneath the painting to find out what the drawing was like, or to see if a painting was a fake. And so I got really interested in this linkage between art history and science and art conservation science. And so and that's how I ended up working at the Smithsonian and working as an art conservator. So it's always linked.
That's very interesting. I have a friend that did a similar path, that was very into like the preservation of art and like the chemistry behind that. So it wasn't something I've ever thought of before.
And I think people will tell you too, just kind of answer an implicit question, that architects and chemists, artists and chemists are really very similar. You've got some idea in your head, you visualize it in your head, and then getting it out there, making it, figuring out how to make it.
Yeah, that's very true. All right. Alyssa, did you have any more questions for Dr. Arkin?
Kind of. So I remember learning a little bit about this, is kind of unrelated, but learning in one of our classes how you would find out if paintings were fake. Did you use like radioactive carbon to do that? Like
Yeah, you can use it. I didn't ever work in that space myself, but yeah, you can use that. You can also, when you understand what pigments, it's totally chemistry. It's actually even mineralogy, which I studied at Caltech a little bit. What minerals are they going to use, and how would they process them, and are those the right minerals? And a particular artist has a very distinctive way of working, right? Like a signature. So you can look at some of the physical nature of the painting to understand if that signature is intact or not. Yeah, it's a cool field.
Very interesting. Yeah. All right, and I say the last question is from Dr. J here at Cal State San Marcos.
So thank you for an excellent talk. So because these are protein-protein interactions, nature has kind of already given a binding surface in the partner in a sense. Is there efforts to kind of do maybe directed evolution to find stronger binding partners and then use those as a guide to developing the fragments?
Oh, yeah, that's interesting. So a couple of different questions there. If especially for those what I was calling primary interfaces. And I can just buy that real quick. Slide 10, I think. 8. So these primary interfaces, you can imagine designing a synthetic molecule based on that kind of a structure. And a lot of work has gone on, especially in these alpha helical types of interfaces, to make synthetic mimics that basically have these fragments. You can think of the amino acid side chains as fragments that you can link in some more drug-like way than a peptide. So then what if I don't have a peptide? What if it's a type of interface that doesn't have this kind of secondary or primary interface? Could I use directed evolution or phage display or some other kind of selection technique to make one and then use that to start my design? So people have done some of that. It's more difficult than it sounds, but it's I think it's another one of those things that could be very powerful. So one of the things that makes that really powerful are people working on backbones, so synthetic backbones that you could in principle apply to any alpha helical system. So the backbone itself, the compound mimics an alpha helix. And then that directed evolution could really be applied to those mimetic backbones. So, thank you.
Yeah.
Dr. J. Actually, Roberto had another question if people don't mind.
Thank you. Okay, I have 2 more questions. All right, so
never mind.
Okay, so the first one, it's really cool that we're on this slide because it kind of revolves around asking about like the complexity of this, but it's like, I guess, do we know what like causes the complexity of these protein-protein interactions? Like, what makes something like primary versus like what, I guess, on the protein or maybe on the whatever is binding, like like why is it like primary versus secondary or tertiary? Like, why is it maybe 2 specific residues versus like an entire beta sheet maybe? Like, do we know why there are tiers of complexity in terms of protein-protein interactions?
Yeah, so let me ask that question in a slightly different way, which is, why did a system evolve to be really simple or really complex? Is there affinity associated with it, or binding kinetics, binding stability? And first, if you look at a very, very broad way, obligate dimers, like proteins that homodimerize, tend to be over large surfaces. Those tend to be tertiary interactions, and extracellular protein-protein interactions tend to be tertiary interactions. And then a lot of signal transduction. So think about this as I said, this is a histone. This is a acetylated histone. So this is a reader domain in epigenetics. So 2 things about that. One, it's a very dynamic interaction. That acetylation can come on and off, and so the BRD4 needs to recognize it or not in a dynamic way because it's in the process of doing transcription. The other thing to it is that BRD4 is not sitting all by itself. It's part of a multi-protein complex. And so as a whole, there are a lot of points of interaction. So those are 2 aspects, I think. And then a lot of the secondary interactions are in signal transduction cascades. So there you can have high affinity, but you can also have rapid kinetics. So this is one way to think about it.
Interesting. Cool. Thank you. Okay, last question. So you had mentioned like using computational stuff. Like, can I ask like what, like we're taking a physical chemistry class right now where we're doing a lot of computing using like charm GUI and like doing molecular dynamics and stuff. Can I ask like what you guys use? Because I'm wondering like how, if like the computers are crazy that you guys are using to make these simulations.
Yeah. So I don't have computers anything like the ones that are necessary to do those calculations. We work with the national labs, like the same computers that maintain the nuclear stockpile. stuff like that. These are like the best computers in the world. And some of the best computer scientists in the world. So there are two basic camps of computational chemistry in drug discovery. One is what you're doing, which is structure driven, like CHARM and molecular docking simulations and molecular dynamics. So those are based on a target and something binding to a target. On the other side, you have data-driven models, and there are a bunch of different, and then you have data-driven models, and those are saying, here's a whole bunch of information. Maybe I don't know the structure. Maybe I don't even know what it's binding to. But from this set of quantitative SAR, structure activity relationships, can you predict what's going to make the compound bind better?
Mm.
And then you do that in parallel over lots of things. So bind to this, but not this. And that's what the computer's trying to do. So sometimes it's sophisticated enough that it can also pull in the docking or the structure-based computation. So far, they don't really help each other that much. They work on different problems, really.
Mm-hmm.
But the goal always is to integrate them to get all the information. Yeah.
Cool. Okay, I'm done. Thank you.
You're welcome. Everybody's saying, I want to have dinner.
All right. Are there any other questions out there in the audience? All right. Well, thank you, Dr. Arkin, for this wonderful talk. And I just want to invite everybody to thank her for attending, or thank you all for attending. Sorry.
Yes, thank you very much for attending. It was lovely to meet you all. Thank you for all your questions. It's, I think, per capita, by far the most engaged audience I've ever had. So thank you very much. This was a great time. And enjoy your evenings, everybody. And you're welcome to keep the video.